Hello viewers, welcome to this channel Anupuma's Biochemistry. This is Dr. P. Mary Anupuma and today I'll be narrating about liver function tests. Liver function tests are a group of tests done to assess the functional capacity of liver as well as any cellular damage to the liver cells. These are done to assess functional capabilities of the liver, such as its synthetic ability, that is, by measuring the various plasma proteins such as albumin and prothrombin that are synthesized by the liver. Also, some lipids which are synthesized in the liver. The next one is its secretory or excretory abilities by measuring the serum bilirubin levels. The common tests done to assess the function of liver are: first one is serum bilirubin estimation. Here we measure both conjugated and unconjugated forms of bilirubin. Then total serum proteins and albumin to globulin ratio. Then assessment of liver enzymes like transaminases and prothrombin time. Here are some enzymes like AST, ALT, and then ALP we can measure, GGT, LDH, all these are the enzymes that we can measure. Next is prothrombin time. Serum bilirubin. Bilirubin is one of the end products of heme metabolism. It is derived from the heme part of hemoglobin molecule and it is an yellow colored pigment. Liver plays key role in its metabolism. After the breakdown of heme portion, it forms unconjugated bilirubin which is insoluble in water. This is transferred from the sites of breakdown, that is from the spleen to the liver by albumin for its conjugation. And in the liver it is conjugated with glucuronic acid with the help of the enzyme glucuronyl transferase. Now we have the conjugated bilirubin which is water soluble and this is excreted into the bile. The normal range of bilirubin is, say for example, total bilirubin, it is 0.2 to 1 milligrams per deciliter. Unconjugated bilirubin will be 0.1 to 0.6 milligrams per deciliter. Then conjugated bilirubin will be 0.1 to 0.4 milligrams per deciliter. Any rise in the bilirubin level to that of 2 milligrams per deciliter results in the symptoms like jaundice, which is marked by deposition of bilirubin in the mucous membranes, imparting yellow color. Jaundice, it is divided into three types depending upon its etiology. We have prehepatic jaundice, hepatic jaundice and post-hepatic jaundice. Prehepatic jaundice. In this case, the cause of jaundice is at the level of bilirubin processing before it reaches the liver. And the most common cause is overproduction of bilirubin due to hemolytic disorders. In this case, the rise in the level of unconjugated bilirubin is more than conjugated bilirubin and hence there is a rise in the total and indirect bilirubin, that is the unconjugated bilirubin will be very high because of this hemolytic disorders. Hepatic jaundice. This is caused by cellular dysfunction of the liver, hence this is also called as hepatocellular jaundice. It is caused by the inability of liver cells to process and excrete the bilirubin in the system. This is seen in hepatitis and cirrhosis of liver, etc. In this jaundice, there is a rise in total, direct as well as indirect bilirubin levels. Post-hepatic jaundice. This is also known as obstructive jaundice as it is caused by obstruction to the outflow of bile resulting in reabsorption of the conjugated bilirubin, and it makes its appearance in the serum. This may be caused by carcinoma at the mouth of the gallbladder or any stone present in the bile duct obstructing its path, the path of flow of bile. In this type of jaundice, we see a total rise in the bilirubin as well as the direct bilirubin levels. Both of them, they rise. Methods of measuring bilirubin. Serum bilirubin level is measured using diazo method that uses diazotized sulfonylic acid to convert bilirubin into azobilirubin, and the color intensity thus obtained is measured colorimetrically at a wavelength of 555 nanometers. Like you can keep the range between 550 to 580 nanometers and measure it. The conjugated bilirubin directly reacts with the diazosulfonylic acid in aqueous medium, and hence we get direct bilirubin concentration. Now its measurement of unconjugated or free bilirubin. So here DMSO is added to make the unconjugated form water soluble and for the reaction to occur. This method, it is the indirect method for measuring bilirubin levels, and it measures both the conjugated as well as the unconjugated bilirubin. So what we get is, using DMSO, the total serum bilirubin levels. Serum albumin and albumin to globulin ratio measurement. Serum albumin is an important serum protein vital for maintaining the plasma oncotic pressure, as well as it acts as a carrier for various biological substances as well as drugs. Albumin is synthesized by the liver, and hence the level of serum albumin, it gives us a stock of the synthetic ability of the liver. Protein malnutrition can cause fall in both serum albumin levels as well as serum globulin levels. The normal total protein level in the serum is 5 to 8.5 grams per deciliter, while the total serum albumin level is 3.5 to 5 grams per deciliter. To get total plasma globulin levels, we subtract the total normal protein levels with the serum albumin level. Then you get the total globulin level, and the range is between 2 to 2.5 grams per deciliter. The normal albumin to globulin ratio is in the range 1.2 to 1.5, and in the case of hepatic dysfunctioning, this ratio is altered. Infective liver or inflammatory pathology is marked by rise in the serum globulin level and a fall in serum albumin levels. Estimation of the albumins using bromocresol green. Serum albumin can be measured using bromocresol green method. Here the albumin, when it combines with bromocresol green in acidic medium to give yellow color, yellow green color or blue green color. This intensity of colors that are obtained, we measure it at 630 nanometers using a UV visible spectrophotometer or a colorimeter. Total protein content is obtained by using biuret method. In this method, the cupric ions of the copper sulfate present in the biuret reagent, they react with the protein to form a violet colored complex in alkaline medium. The intensity of this violet color, it is measured at a wavelength of 540 nanometers. Prothrombin time. Prothrombin, as we know, it's a clotting factor 2, and it is one of the important component of both intrinsic as well as extrinsic pathway of blood coagulation. This prothrombin, it's a serine protease. It's a serine peptidase which converts fibrinogen to fibrin. This prothrombin, it is synthesized in the liver. Hence, prothrombin activity in plasma that can be used to measure the synthetic function of liver. Determination of prothrombin time. First, we have to collect the blood in tubes containing the anticoagulant citrate. From the blood, we have to isolate plasma. Then we have to place the plasma in an automated machine that adds excess of calcium to reverse the anticoagulant effect of the citrate that we have added. And it measures the time taken for the fibrinogen to be converted to fibrin, and hence measures the activity of thrombin present in the plasma. So the time duration taken gives us the prothrombin time. The prothrombin time differs in accordance to the analytical method that is used. Hence, to compensate for this, international normalized ratio is used. In this, the manufacturers of the kit, they assign an international sensitivity index value. So this shows the amount of tissue factor present in the kit as against an international accepted standard. The SI value is generally 1 to 2. Now this method can be used wherein the prothrombin for clotting time for the test sample versus control is done, and the ratio, it should be between 0.8 to 1.2. This is considered to be normal for patients who are not taking any medication like warfarin. And in case of individuals who are consuming this particular medicine, then for any other disorder, then the value will be around 2 to 3. Liver enzymes. These are most commonly used to assess the liver function tests, and these enzymes, they are of hepatic origin and usually they are not leaked. They will be leaked only when there is destruction to the hepatic cells. So liver enzymes are measured to get an idea of the cellular functioning of the liver, and if the value is increased because of any condition such as viral infections, that is in the case of viral hepatitis, toxic hepatitis or cirrhosis of liver, then we can say that there is some problem with liver and some infections are there. So that's why they measure this liver enzymes. Usually they are not leaked. They will be leaked only upon destruction of hepatic cells. So some enzymes which are measured include the transaminases, the transpeptidases and the phosphatases. We shall see them in detail. First is transaminases. Transaminases means these are the enzymes that transfer amino group from an amino acid to a keto acid of another amino acid. So these are used in the interconversion of a keto acid to an amino acid and amino acid to a keto acid. Transaminases are measured to assess the liver functioning test, and these include the alanine transamerases and the aspartate transaminases. These two enzymes are measured. Alanine transaminases, they catalyze the following reaction, like alanine plus alpha ketoglutarate will result in pyruvate and glutamate. So amino group from alanine is transferred to alpha ketoglutarate to get glutamate, and alanine, as it loses amino group, it becomes pyruvate. Same is the case with aspartate transaminases. This particular enzyme, it transfers amino group from aspartate to alpha ketoglutarate, and this will result in the formation of oxaloacetate and glutamate. The normal levels of alanine transaminase in the serum is 7 to 140 international units per liter, while the normal levels of AST is 8 to 40 international units. And any increase in AST or ALT levels, it hints a damage to the liver parenchymal tissue. And if I have to say, ALT is more specific marker for hepatic injury. And elevation in AST is also seen in cardiac tissue injury, hemolysis, and muscle tissue damage. UV method is used to assay these things, and it is coupled to a reaction in which NADH is used, resulting in the change in the photometric intensity when it is read in the range of 340 nanometers. And it is a UV kinetic method. So as the enzyme is acting upon the substrate, NADH is consumed, thereby resulting in change. See here, alanine plus alpha ketoglutarate gives rise to pyruvate and glutamate, while pyruvate, which is the product formed here, combines with NADH, consuming the NADH, resulting in lactate and NADH. So the more the pyruvate formed, the more the NADH consumption, and you can see the color intensity change. Same is the case with AST, wherein aspartate is converted to oxaloacetate, and oxaloacetate gets reduced by NAD+ to form malate. So this kinetic conversion will lead to a change in the OD. Alkaline phosphatases. Phosphatases, these are hydrolases that remove the phosphate groups from any kind of molecules, like from the proteins, even nucleotides, etc. And these phosphatases, they are found in cells lining the biliary system. Hence, a rise in the level is an indicative of damage to the biliary tree due to cholestasis. This may also be due to stone blocking of the large ducts, or intrahepatic obstruction, or inflammation of biliary channels. Alkaline phosphatase is also found in the placenta and bones. Hence, the level is also increased in growing children, in whom these bones they undergo remodeling, and in Paget disease in case of adults. So in Paget disease, old pieces of bones, they are replaced with new ones in the adults. So in these situations also you find elevated levels of alkaline phosphatases. Normal level of alkaline phosphatase is between 45 to 115 international units per liter. And the measurement is by kinetic method. So it uses para-nitrophenyl phosphate as the substrate for measuring of the enzyme, and the measuring rate of formation of this colored end product, that is para-nitrophenol, is formed. The rate at which this is formed indicates the concentration of the enzyme that is present. This measurement of color intensity is done at a wavelength of 405 nanometers. So here the para-nitrophenyl phosphate in the presence of ALP, alkaline phosphatases, in the presence of water, is converted into para-nitrophenol and phosphate is released. So the increase in intensity of this color is directly proportional to the concentration of the enzyme. Gamma glutamyl transpeptidase. This enzyme is specific to the biliary tree again, and a more specific indicator of cholestasis and damage to the biliary tree. It is also a high specific marker and is raised in even minutes, and subclinical damage to the biliary tree can be assessed. The normal range is between 0 to 42 international units per liter. Thank you for watching this video. Please do subscribe our channel for further videos.